Bioinformatics.
Okay, so welcome to another lab follow along. This one is going to be on the variant calling lab, which was kind of the second part of the read mapping lab series, essentially. So again, this is not intended for you to follow all the way along through, start to finish. This is meant to be kind of a resource for you to skip around and see how I did some of these things and see how I typed it and maybe what I have to say about it. So again, totally optional resource for you. So diving right in, our goals today are to do something we've already done before, which are to map reads to a reference. And then we want to convert between a SAM and a BAM file, something you haven't done. But I did mention in lecture that SAM and BAM are readily interchangeable, and really you just change them based on what the program you're planning to use requires. And then finally, we're going to call SNPs or SNVs, depending on the frequency in nature. These are basically single nucleotide polymorphisms. We're trying to find places where our sequenced organism, when you map its reads back to the reference organism, where there are base pair substitutions. And that's really what we're going to try and get out of this lab. So to get started, let's go ahead and make a directory. And so we'll just call this lab 10, and we'll cd into lab 10. And then I've got a couple things that we're going to download. We're going to be doing some stuff with E. coli in this lab. And so we're going to download an E. coli reference genome, which we will be mapping to. And what we will be mapping to it are some raw E. coli reads. These are FASTQ. We're not going to do any quality control on them today because you've already learned how to do that. In a real scenario, you would though. And then we're going to unzip the reference as well. So I'm going to copy and paste these first two things so I don't have to worry about mistyping the URL. And it's unzipped. If we type ls, we can see the references right there. So let's go ahead and FASTQ dump all of these reads. We're not going to split them or anything like that. These are single end reads to make things nice and simple. And once that's done downloading, we'll go ahead and make a directory for our reference so we can stay a little more organized. As you recall last time, when you index references for the purposes of mapping, you start to get lots and lots of files. So I like to confine all of that into its own directory. So we will make dir reference, and then let's go ahead and make use of that move command, mv, and we'll move our E. coli reference into reference. And so if we ls reference, we'll see that it's in there now. We've started to explore the use of environmental variables to kind of make our lives a little easier with some of these long file names. So we're going to go ahead and kind of build on that more and actually use our environmental variables to also help name our output files as well. And you'll see how that works as we move forward. But first, let's go ahead and provide a absolute path to our reference. So it's the home directory /lab10/ref/E. coli. And let's provide the absolute path to our reads as well. So again, lab 10, only these are in our base directory of lab 10. Okay, so you could go ahead and head read if you want, just to verify that you indeed got it. And I'm going to trust that I did reference correctly. So we have done this read mapping before last time, right? So I'm not going to dwell on it. We're just going to kind of power through it and get on to how to actually use the mapped reads this time. So we're just going to be using BWA this time, and we're going to index our reference. And then we will map our reads onto our now indexed reference using BWA mem. And here's where we can use the environmental variables to make our output names nice too. We're going to name it the same thing as the variable name for the actual reads themselves, only we're going to append .sam to the end of it. And so this also maintains all the pathing and everything as well. So this is quite a nice way to do it. And so when this is done, I'll go ahead and okay, so once your read mapping is done with BWA, you're pretty much now at the point of where we got to in the last lab. Only this time it just took a few minutes. So next what we need to do is we're going to start actually having to play with these a lot more. And first I want to show you how to explore the mapped reads a little bit so we can look at how they did, what's mapping, what's not mapping a little more clearly than just the output from BWA or Bowtie 2 like we did last time. And so we're going to use Samtools, and before we use Samtools to start looking at these things, Samtools actually also wants you to reference the index in its own special way. So without going into too much detail, we need to index the reference one more time. Now, this one doesn't tell you anything. It's what we call not being very verbose. But if you were to ls references, you could see that there's actually some new files in there as well that were just added by the Samtools indexing. So everything we're going to do here forward is going to want a BAM file instead of a SAM file. If we go ahead and head our read.sam, you can see that again, it's human readable. Now, it's no longer going to be human readable because we're going to further compress it by converting it to BAM. However, now that we've indexed it with Samtools, it's really quite easy to use the function Samtools view -b big S. Now what this stands for is we want a SAM, which is the lower b, and the big S means that we have a SAM. We could invert that and say big B little s if we wanted to go the other direction. So we'll go read.sam. And notice I hit tab to tab complete because that's a habit of mine. It actually filled in what the actual environmental variable is standing in for as well. That home/ubuntu/lab10 blah, 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 blah. If I didn't hit tab and it still said $read.sam, that's fine as well. I just have a habit of mashing the tab key. And then we'll go read.bam as our output. And we'll hit enter. And then we need to do one more thing to our BAM file. Most things that are working with mapped reads want the mapped reads in some semblance of organization. Typically, your raw reads don't really, when you first get them from the sequencer, don't come in the order that they appeared in the genome, right? Because they're all pieces sequenced at random. And when you map them, your SAM file, they're in an order that just reflects the order that they were in your sequence file to begin with. And so what we want to do is we actually want to sort it in a way that makes things more efficient. We want things that map to the beginning of your sequence to come at the top of the SAM or BAM file, and the things at the end to come at the end. This kind of helps things when we start to stack them up, as you'll see. So we need to do Samtools sort on our BAM file, read.bam. And by my own convention, I always name these things with a .sorted before the BAM now. So now that I know that this is the sorted version, and we'll hit enter. While this is running, I'll just mention, you now have all the pieces if you wanted to go back and just visualize these and go, okay, well, let's see how these reads mapped, how they actually look. We played around potentially if you wanted to do that optional part with tablet last time. You can take that E. coli reference and your SAM or BAM files and import these into tablet as well if you wanted to kind of scroll around and look at them as we do some of the things coming up as well. Looking for the SNPs and SNVs and seeing if you can identify them using that SNP slider as well that we saw some of the forced mutation from our simulated data set. Okay, so now we're going to use Samtools view to start and get some information. And this is just barely dipping our toes in the water. There is so much information in these SAM and BAM files that you could go crazy. We're just going to look at one aspect of that flag column. If you recall, that flag column is that really weird number that's associated with 2 to different exponent numbers as true-false statements for whether something has a pair, whether it mapped, whether it was unmapped, whether it was on the one strand or the other strand. So we're just going to keep this very simple. Let's say we want to see which reads mapped versus which reads didn't map. Now, when I would use this out in the real world is not only seeing how my alignment performed or my mapping performed, but let's say I was looking at plant DNA or plant RNA, something that's easy to contaminate, and I knew that a human was working on it. One of the first things I might do is actually map all my reads to a plant genome and then pull out all the unmapped reads and pull out those unmapped reads and then see if they map to a human genome to get a sense for how much contamination there potentially was. And so this is kind of the types of things we can do with Samtools view and some of the flag stats as well. So the flag stat for things that are unmapped is a 4. And I've got links here if you want to go see all the different things that you could pull out. So we'll go Samtools view -c so we get a count of how many things it was. We could also use this to pull out the actual sequences as well, but we just want the count right now. So read.bam, and here you could also use the sorted BAM, but this part doesn't require it. We'll be using that a little bit later. And here we've got around 334,000 reads were actually not mapped. Now that doesn't mean there's anything wrong with the mapping. It could be contamination, like I mentioned. So But, you know, where's the context? How many reads did we start with? So lots of different ways to think about how many reads you have. But why don't we just go to the source, and why don't we cat our read.sam file if you wanted to figure out how many. And remember, every single line is a different read. So we could word count -l for the lines, and we could see that there's actually 7 million reads. So 300,000 is still a pretty decent chunk of that, but not half of it or anything like that. So it would depend on what you were really trying to do for whether that was an unexpected result. Okay, so that's kind of, you can play with that and those flag stats, and I encourage you to play with it. But next, if we want to have a little more specific output, we can also look at Picard tools. And so we're going to use Conda to install Picard. Now, Picard tools has a ton of different independent tools all within it. We're just going to look at one, which is collecting some basic summary metrics from our mapping or alignment. And if you wanted to see all of them, you could just execute Picard by itself. And here we could scroll through and see there's VCF converters and all sorts of stuff that probably doesn't make sense right now. SAM to FASTQ. If you ever wanted to pull out all the FASTQ files just from your SAM file, that could be very useful. If you found all those reads that were unmapped and you were worried about them being contamination, you could get them back to being a FASTQ and do all sorts of stuff with them. So lots of really helpful things within Picard depending on what you're trying to do with your SAM files. But for right now, we're going to use collect alignment summary metrics. And so we're going to go Picard, collect, and you can't tab complete this part. So I apologize if I end up mistyping it here. Metrics. And R is going to equal our ref. I is going to equal our read. Dot, this is where we need the sorted one. Dot bam. And big O, not a 0. That'll actually not work for you. We're going to call our output statistics, and I probably spelled it wrong, dot text. And you can see I actually had a typo right here. Sorry about that. And so now this is going to run for a few minutes. All right, so now that that's finished running, you could, if you wanted to, you could cat the statistics file that you had as an output. Now, I'll warn you, at first it looks kind of normal down at the bottom, but actually that's because it's wrapping around. This is actually a table with basically one column in it because we have one set of reads that we mapped into this. You could map multiple files and you'd get multiple rows. These are basically tons and tons of columns, but because it's really long and it's got a lot of, it's spaced with tabs, it's not great to look at within the terminal. You could go ahead and use CyberDuck and download this and open it in Excel or something else as a tab delimited table as well. But you could also go ahead and click on this link that I provided with you where it shows what some of these columns are and what the associated values mean. So like mean read length of the reads within your mapped reads, things like that. Percent of things that are chimeric, meaning that they map to 2 different chromosomes, things like that. So you could kind of dive deep on this. Just know that these pieces of information are here if that's ever something you're interested in. Okay, but we're really here to identify where are the single nucleotide polymorphisms. So let's go ahead and install one more thing. I think this might be the very last thing I have you install today. So we're going to go through Bioconda and get BCF tools. Okay, so we've kind of installed a lot of different tools today. Now, we're going to do 2 things here. We're going to use BCF tools to implement mPileUp, and then we're going to call the SNPs. So what mPileUp does is it takes all those little reads along that are mapped on top of your reference. And again, you kind of saw that when you use tablet and look. And it actually stacks them all up. So reads that hit a single base pair, stacks all of them up, and it looks down all of them and says, what number of these reads have an A, which have a T, which have a G, which have a C? What's the ratio between them? And so we, instead of saving that output, which you could do, we pipe it immediately. The output, rather than going to the terminal, goes to BCF tools, where it calls now, based on some criteria that we give it, whether it thinks the genotype there is homozygous A, whether it thinks it's homozygous G, whether it's a percent likelihood of it being heterozygous AT or AG, whatever. And you can also specify the ploidal level so it has an idea of how many potential alleles to expect at any given site. A tetraploid could have up to 4, for example. And so you can play around with all these things. I'm going to start executing it because it can take a while, just like everything we're doing right now, even with a small data set. We're getting into the types of analyses that when you use real data can take hours, if not days. Some things can take weeks. And so while this is running, I'll just mention on the note of the heterozygosity, if you saw 50-50 of all your reads over a given site showing half showing G, half showing T, you'd be pretty certain that it was heterozygous. But what if it was 40-60 or 30-70? That's why we implement not just a is it heterozygous or not, we implement a likelihood. What's the likelihood of it being homozygous or heterozygous at that site? Okay, so I'm going to let this run and then come back. Okay, so it finally finished. Again, this one probably took a little while. And let's go ahead and use tail to look at the bottom of our VCF file. Now, if you don't remember the name at the end of it, why don't we use wildcards, because we know there's only going to be one VCF file in this folder, and go ahead and look at it. Now again, this is another kind of table-based format. Every row is going to, kind of wrap around here, but we've got different things in here. So we've got the species, the genome position, we've got what the base was in the reference, what it is called as a polymorphism, and then all these weird kind of values. VDB equals 0.93, blah, 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 blah. Now, this is highly customizable. You can figure out what types of information you actually want to pull down when you create your VCF file. These are basically different types of stats and things like that. You could read more about the VCF format to find out that, but if you just wanted a quick answer, what I like to do is, because if you were to head this file, so star.vcf, you've got all this info on all these different things that you've asked for, like IDV is the maximum number of reads supporting an indel, all sorts of things. So rather than scroll through this and read it, why don't we just work smarter and use grep? And we could grep for ID equals whatever metric we're looking for, kind of to be unabbreviated. So if we're interested in the AC metric, we could just grep our read.vcf file for that. And here we can see from that intro sets of lines, AC, the allele count in genotypes for each alternative allele in the same order is listed. Okay, and so that gives you a little bit of information about what that metric is representing. And you could do this. I encourage you, play around, look at kind of the tail output. Think about what is MQSB, what's SGB. What are some of these pieces of information that we're getting? All right, so we've got our VCF file. It's got where all of our SNPs are located and information on what reads they came from and how they differ from the reference and how confident we are in all these different things. And so let's take one last step and let's actually incorporate some biological information now, because we're just thinking about letters right now. But where are these SNPs, and are they in genes? And if they are, what are these genes doing? And this is where incorporating annotations can be very helpful. So we'll go ahead and wget a annotation for our E. coli reference and unzip it. And so an annotation file, if you want to head it, it's in .gff. And what this is basically an annotation of is everything that we know about the different regions in the genome, whether it's a gene, whether it's protein coding, all sorts of things, and its location. So we can combine this with the reference, the reads mapped on top, and then the annotations on top of the reference, so that we not only have locations of where these reads map and SNPs map, but what functions they're involved in as well. And this is where it starts to be informative. So we need to go ahead and install Bedtools if we haven't already. So I'm just going to copy and paste this, and I'll put a -y on the end to make it say yes automatically, which I sometimes forget to do. And what we're going to do is we're going to start looking for intersections between our annotated reference as well as our VCF file. And so this can get a little confusing, and I've got a link here if you want to read more about it. But basically, there's two ways from a simple example. There's two ways of thinking about how to ask for overlap. So if we think about reads as being in this kind of tan part right here, and our reference being blue. So let's say our reference A is here, and each of these blue boxes represent a gene on top of our reference, an annotated gene. And these, again, these yellow spots are reads. What we want really are to pull out pieces of information where we have both of the blue and this tan present. Again, these are the ones that have SNPs contained. So we could ask for the intersection of A and B, where it's just the pieces where both are present, or we could ask for places where they're not both present, or we could ask for the entirety of the annotation from the reference that has at least one of these pieces overlaying on top of it somewhere. And that's really the one I like to get, because we don't want just the piece where the SNP is, right? We just say, give us the whole gene, and then we can look and go, okay, there's SNPs in this gene. So if we do this argument WA, this is how it looks for the intersection. Now it gets more complicated, and I don't want to get too in the weeds in it. You're welcome to kind of look at it. Just know that if you want just it based on the read overlap alone, you would not apply this -WA argument. If you want the entire gene, regardless of where the SNPs are on that gene, you apply the WA argument. And so here, that's what we're doing. We are saying Bedtools intersect our A, which is our annotations, the E. coli annotation, and B is our read.vcf. So again, the intersection of A and B, and we're saying -WA, which is going to give us all of the A annotation anytime there's a SNP on that. And so we'll go ahead and pass this through. What you'll notice is this is going to all just rapidly print to the terminal, okay? And so we can see all that information from the annotations where there were SNPs. You could redirect this and call this SNP genes or something, .text, and save it if you really wanted to. But why don't we just, look at how many annotations we pulled out from our SNPs. So something we could do is we could pipe this to WC-L again. See that there's 381 annotations that have a SNP in them. We could also pipe this to grep if we want more specific information. So let's say I knew that the species I was studying right now had overactive membrane proteins. We would expect there to be SNPs there. So why don't we just grep this output for membrane protein? Again, this is a very simplified example. But we can pull this out and say, okay, well, here's all the genes, and you can see the GenBank ID if that's something that you're interested in, where you could go and look more closely at this gene, or you could even go pull this back out from the reference as well. So you could play around with this. You could even get the inverse. Again, I showed you that you can play around with how the intersection is done. So you could find things that don't have SNPs, things that are conserved. You could also play around with your grep output too. So what if you wanted everything that's not a membrane protein? So, you could play around with whether grep's an inverse search or not as well. We've played around with that with the -V argument. And then you could take all this as well, and you could input this into Tablet and look at this. You could play with that. We've already done a little bit with Tablet, so, I consider this optional. Again, sometimes it's fun just to visualize things in colorful ways. And if you really are feeling like you're chomping at the bit for more, because maybe this was a shorter lab, you are more than welcome to kind of read what I've put down here. I've kind of outlined kind of a do-it-yourself project for it, where I'm providing a reference genome for yeast as well as the annotations. Again, this is a bigger genome, so things will take a lot longer. So I would say you could skim the SRA and find some yeast sequence data if you wanted to do it totally from scratch and see what kind of SNPs are there relative to the reference. Or you could look at the one that I used, which was a iron-resistant strain of yeast. I was interested to see if there were SNPs within genes related to iron resistance. So that's something you could look at. I will say only deal with around 200,000 reads, so make sure you subset your FASTQ dumps. Otherwise, you'll be there for hours. Okay? And so if you don't do that, that's fine too. Just trying to give you kind of tools for kind of expanding if that's something that you're interested in. All right, thank you.